the PowerPoint, this one, of the chapter 24, biosynthesis of amino acids, which we have gone through last week. And let us see, this is the root nodules of where the nitrogen fixing bacteria, rhizobium, which is accumulating over there and fixing the atmospheric nitrogen. And that's the part how the amino acids biosynthesis, that entirely depending upon the availability of nitrogen, and that nitrogen should be fixed only by bacteria, not by plant, not by animal. So the animals and plants, they depend upon the amino acids or nitrogen fixed by bacteria. So in that sense, the evolutionary perspective, first in the evolutionary process, the earth started, and for that you need of all gases and oxygen, hydrogen, nitrogen, all the other gases, and then when the life starts, actually you have to have some enzyme formations. In that sense, it will fix the atmospheric nitrogen into ammonia, and then a lot of ammonia, and then next level is use this ammonia by the plant, and then consumers of the plant is the animals, and consumer of animals is the human being. So it's going on and on. That is the evolutionary process. Okay, so it is a theoretical possibility. There is no one is ever proved this is right or that is right. So as per the mathematical relationship, yes, this in add to another one, you get 1 plus 1, you get 2, 2 plus 1, 3, 3 plus 1, 4, 4 plus 1, 5. Like that, the mathematical relationship. Like that, people, they build one upon the other. In that sense, the amino acid synthesis is coming from ammonia, and ammonia is coming from nitrogen, and nitrogen is coming from atmosphere. So that is the changes. And these are some of the the reaction which we saw last class. The nitrogen and the nitrogen bond is one nitrogen atom, another nitrogen atom. It is being bonded by triple bond nitrogen, and this is very hard to break. So in a physiological sense or physical sense, you need a lightning, a power of lightning, the energy that will break this bond, and then hydrogen which is present in the atmosphere can join together on ammonia. This is a type of reaction which is naturally occurring. At the same time, this nitrogen the bond can be broken by the nitrogenase enzymes from the bacteria, and they can also produce the ammonia, and this ammonia is converted to glutamate, and glutamate is being used by the organisms or the bacteria, or from the plant, and or from the animal systems too. And thereby it helps to synthesize the amino acids. And if you go through this electron reductase, the enzyme which is responsible for the fixing the nitrogen is for the iron protein, that is reductase, a nitrogenase enzyme. This is a molybdenum and iron protein. It is an iron protein enzyme. And this is the way how this energy is being used and split up the nitrogen molecule into nitrogen atom and to form ammonia. So need more of energy. And this is by enzymatic process. And this is again the mechanism of this particular enzyme. Cartoon. So you have carbonyl compound, amino donor, and Schiff base and protonated Schiff base. So you remember the Schiff base is the basic mechanism of the synthesis of the amino acids. So here the ammonia donor in one side, and then the carbonyl compound, C double bond O, carbonyl. And then look at this, is being changed by R3N here. And then hydrolysis, you have RNH, and then R to protonated Schiff base. C double bond N, that is actually a Schiff base. So you have to remember some mechanism in the synthesis of amino acids. It should go through the Schiff base, C double bond N. And then alpha ketogluteric acid, C double bond N. Here you can see that, and then the glutamate is being formed. This is the mechanism. And this is again the sticky and the oxaloacetate, one of the TCA cycle product, or one of the intermediate, how that is involved in the synthesis of different amino acids. And this is again how the phenylalanine tyrosine tryptophan pathway. Pyruvate is the origin for the alanine, valine, and leucine. Ribose 5-phosphate that gives to histidine. And alpha ketoglutarate to form glutamate, and glutamine and proline and arginine. Okay. Serine, hydroxy amino acids, that is derived from 3-phosphoglycerate that forms the cysteine and glycine. And these are the non-essential and essential amino acids. And you can have the list, and you can also the number of amino acids, the number of steps in pathway. These are some of the enzyme mechanism. These are the enzyme linked to it, how this enzyme reaction which is taking place from glutamate to alpha ketogluteric acid. So I'm not going through all those PowerPoints, and please go ahead in your text book, as well as I posted in the blackboard, so you can go through and use my PowerPoint, my document Panapto lectures of the last class. And one of the important steps is the S-adenosyl methionine, or we call it as SAM, that is involved in the active methyl donor. So this S-adenosyl methionine to form S-adenosyl homocysteine, and then homocysteine, and then methionine. And this is a cyclic process where the S-adenosyl methionine is a methyl donor. Okay, so that's another active group. And oligonucleotide target base to be methylated. This is again the mechanism of the enzyme crystallized structure. And here how this will be activated, SAM reaction. This is another pathway, chorismate and glyphosate roundup, which I explained last class. This is tyrosine and phenylalanine formation from chorismate. Then this is the tryptophan synthesis. Then here tryptophan again. Phase. That's again feedback inhibition mechanism, how one particular amino acid is being regulated by feedback inhibition. The product A to B conversion by enzyme, B to C, C to D, D to E, and E to Z. But the Z, the end product Z, the biological reactions won't allow the accumulation of all the amino acids in one direction. Some part they need to regulate by the product of ZC, or the end product will go and inhibit the first product conversion from A to B. So this is the feedback inhibition. Its own product will inhibit its synthesis. Okay. And these are the other mechanisms and how the feedback inhibition, isoleucine and valine. Here the regulator sensitivity, and then this is again the reactions with de-adenylated glutamine synthetase to adenylated glutamine synthetase. So how with phosphorylation, one is activated and another one is inactivated. This is the glutamine UMP-UTP pathway. And you have the selenosulfate, selenic acid, selenate, synthesis of arginine and N-hydroxyarginine citrulline synthesis, and that how the nitric oxide and urea formation. And here one other thing is the heme synthesis. Succinyl-CoA and glycine is involved in delta amino levulinate, and this levulinate form a ring-like structure to form the heme biosynthesis. You can see this one. 8 molecules of delta amino levulinate to form a ring structure, the linear, and then the uroporphinogen and coproporphinogen, and then protoporphyrin 9 with the iron, it forms the heme synthesis. And these are some of the reactions. And then the heme product, the breakdown product of heme become biliverdin and bilirubin. These are the pigments, and this is normally exists in our body in the blood, but in a hemolytic jaundice and some of the infection in liver, viral infection where the heme RBC is broken down or heme is broken down, this process has been highly activated, and thereby you get excess of bilirubin, and that will induces the yellow color and everything, and urine is being excreted enormous amount of bilirubin. Tryptophan, that the degradation ornithine and proline, bromoethanolamine, histidine, and these are the some other product which is the experimental way that you can prove light adapted, dark adapted, and how these amino acids being changed to the level. So this is for the last week's class. So just I want to recapture what we have left, and today's work we have to go in this one. That's on chapter 25. Here you will find the chromosome structures, and we are going to study about the nucleic acids or nucleotide biosynthesis. Last class we did the amino acid biosynthesis, and the nucleotide biosynthesis is directly or indirectly involved in the amino acids as well. We are going to see one by one today. And this cartoon, this photo micrograph, the yellow color is the fluorescent structure. There you get the dihydrofolate reductase enzyme, and this enzyme is being inhibited for methotrexate, an inhibitor, anti-cancer drug, which will inhibit the chromosome division. In order to inhibit it, inhibit the reductase enzyme, and that because of that inhibition, so this particular gene is highly expressed, and it is not being, is being used much because that enzyme is being inhibited. So the accumulation of the gene product, and as well as the expression of the gene, which is being shown onto this chromosome as a yellow color. So the response of methotrexate, this is also expressed in your textbook picture. And here you can see the methotrexate is a drug, and that is an anti-cancer drug, which is this enzyme is a reductase enzyme, where NAD, and it binds in this enzyme and inhibits. So that's the cause. Okay. So let us go further into our topic of today's work. Could you see now from Victoria or the document camera?
Yes, sir.
Yes, okay. Great. So we can go for the chapter 25. Could you see something on it? Chapter 25. Okay. Nucleotide biosynthesis. Okay. Nucleotide, which is required for what? Nucleotide required for cell growth. Is that right? Cell growth. In other words, also cell or chromosomal replication process. Okay. So the key enzyme for this the biosynthesis is dihydrofolate reductase enzyme. So this is one example which I just showed you onto this one, the PowerPoint which I showed you, that enzyme. And this is inhibitor for this methotrexate. This is an anti-cancer drug which will inhibit this enzyme. Why? Because this enzyme is responsible for the cell growth as well as cell replication. In other words, there are different steps involved in the cell replication. One of the steps is a dihydrofolate reductase enzyme is participating in the cell replication or cell growth. So in the cancer cells, which is uncontrolled cell growth or uncontrolled mitosis or uncontrolled replication, that can be inhibited by the methotrexate, which is a key inhibitor of this particular enzyme. So the picture which I showed you is the fluorescent micrograph of the methotrexate treated cells, and where the cells contains hundreds of copies of reductase gene that is being activated. Okay, then what is the regulation? Now we have to know about the regulation, or what is the role of the nucleotide, regulation of nucleotide. First, we should know about the role of nucleotide, different role. What is the function of this nucleotide, role of nucleotides? And what is the nucleotide? What is a nucleotide, and what all the nucleotides have been available? And how these nucleotides participate in different reactions, and then what is the synthesis? That's what we are going to see today. Okay. Number one, nucleotides are the activated precursors. Activated precursors of nucleic acid. You know what is nucleic acids? You should know about by this time DNA and RNA, deoxyribonucleic acids and ribonucleic acids. So, nucleic acids necessary for nucleotides. Again, they are nucleotides. I put NT abbreviation as a nucleotide. Okay, NT nucleotide, they are necessary for cell division. Okay, or replication of a genome. Not only the bacterial genome or plant genome or animal genome or any genome. Okay, they are necessary because they are the building blocks of nucleic acids. And also nucleotides is necessary for the transcription. You know what is transcription? By this time you know about the transcription of genetic information. What are those? RNA formation. Okay, ribonucleic acid. Transcription of genetic information. That's the RNA formation. And also nucleotides is important as an adenine nucleotide, for example. Adenine nucleotide. Adenine nucleotide is involved in as ATP. As you know, they are energy currency of the cell. Okay, energy currency of the cell. So the next one, the GTP, or the guanine. This is the guanine nucleotide. Guanine nucleotide or GTP. They are also energy resources. Energy source for many reactions. Okay. Then the nucleotide derivatives. So not only the nucleotide alone, but nucleotide derived derivatives like nucleotide derivatives. nucleotide derivatives. For example, UDP glucose. UDP glucose, the UDP uridyl diphosphate linked with the glucose molecule, they participate in glycogen synthesis. Okay, involves in glycogen synthesis. Glycogen synthesis. Okay. And nucleotides, they are the essential components of nucleotides are the essential components of signal transduction pathway. Signal transduction. Energy signal transduction pathway. Could you see now, or from the document camera, or it is not clear? Could you see from that one or not? No? Or I have to change the pen? Is it okay? Could you see from your screen? Let me turn off this on or off. No, this is not the thing. Okay. All right, I'll change the pen. That's the ideal thing. I'll do that one. Also getting glare on that. Okay. So the nucleotide, which is involved in the essential component of this signal transduction pathway, and that's on the cyclic AMP, and cyclic GMP. Both of them are acting as the second messengers. In a signal transduction pathway that transmit signals. Where it transmit the signals? Transmit the signals within and between cell and between. See, one cell to other cell, between the cell, and within the cell it can also transduct the pathway inside the cell. So it's within the cell and this is the between the cell, so it can go through. So the cyclic AMP, cyclic GMP can act on both sides, within the cell or between the cell. So nucleotides are involved in the transmitting the signals from within the cell or between the cell. So it plays a major role, okay, in signal transduction pathway. Now the next work of the role of the nucleotide is the ATP can also act as a donor of what? Phosphate group. Donor of phosphate group or the protein kinase for the reaction for protein kinase reaction. Always the kinase reaction involves the presence of ATP. So ATP also a nucleotide that involves in the phosphate transfer group or to the protein kinase. So it also activates the other molecule. Okay. In some instances, the ATP also secreted from cells, as signal molecule. If ATP is secreted outside the cell, that means there is a signal, which is a signal molecule for the other. So we have covered most of this role of nucleotides, and now shall we go into the biosynthesis of these nucleotides. Okay. The biosynthesis of amino acid, just now we have seen the same way these amino acids also which is involved in this biosynthesis of nucleic acid. Biosynthesis of nucleic acid. I put nucleic acid. It involves, nucleic acids means it has a nucleotides, right? Nucleotides. And the nucleotides is made up of what? Ribose or deoxyribose in case of DNA, as well as deoxyribose plus a base like adenine or guanine or cytosine or uracil, and also thymidine. Okay, any one of them. Just it involves like either DNA or RNA. You have a deoxy ribose here or ribose here, but the adenine, guanine, cytosine and uracil are present in the RNA, but A, G, C, T for DNA. Okay, plus a phosphate group. So all of them, this is number 1 and this is number 2 and this is number 3, all of them constitute, we call it as one nucleotide. Like a ATP is a nucleotide, all of them, triphosphate. AMP, monophosphate, also a nucleotide. Okay, and ADP, that is also a nucleotide, the diphosphate group. So like that you have a GTP, GMP, GDP, like that. You know, it goes on and on. And if you have a deoxyribose, so we call it as a small d, DATP, or DAMP, or DATP, or DGTP. So deoxyribose. If you are ribose, you have ATP, but deoxy, you have to say DATP. That's the respective nucleotide. These are some of the nomenclature which can also be seen in the table. I'll show you a little bit. Like amino acid biosynthesis, here in the nucleotide scaffold, I call it as a nucleotide scaffold, because the different molecules or different atoms is coming from various origins. Okay, the assembly of atoms. Scaffold means, you know, it's being assembled from with the different atoms and derived from different compounds. So the assembly, how they are assembled, the assembling of the atom, assembling of atoms of nucleotides. That's very important. Which atom is coming from where? Which amino acid or which compound, which molecule? So one example here is, you can say glycine is amino acid, and aspartic acid or aspartate is another amino acid. So they contribute, suppose in a hydrogen or carbon, and some of them are acid group, some of the nitrogen is coming from here, you know, in the synthesis of the side chain. The ammonia group, amine group, which is coming from ammonium, NH2 amine group, which is coming from glutamine. So these are some of the contributions. You know, each amino acid will contribute 1 or 2 atoms for the synthesis of nucleotide. Okay, so we will see how they are assembled. That's the topic today. That's the biosynthesis of nucleotide. Okay. So in table 25.1, that's onto your PowerPoint. We will see here. Okay, here. Nomenclature of basis nucleoside or nucleotide. Could you see that? Okay. Adenine, guanine, uracil and cytosine, these are the base for the RNA, and the ribonucleoside, adenosine, guanosine, uridine and cytidine. This is with the sugar alone, and you get the phosphate group is added, then they call it adenylate, guanylate and uridylate and cytidylate. The same thing which is going on here, adenine, everything is the same but except the thymine. But here you got a deoxyribonucleoside. So you got deoxyadenosine, deoxyguanosine, it's not deoxy, but thymidine here and deoxycytidine. And then you can go here, thymidylate, deoxycytidylate, deoxyguanylate, and then deoxyadenylate, DAMP, DGMP, and TMP. It's not DTMP, but it's the TMP, thymidylate. And here DCMP, because this is the unique one when compared to here. So, let us go, these nucleotides, they are synthesized through two different pathways, and the pathways which is called as one pathway is salvage pathway, another one pathway is de novo pathway. The salvage pathway is an activated ribose, phosphoribosyl pyrophosphate, PRPP, plus a base, direct add. So this base is coming from already made or the recycling, and thereby nucleotide can be synthesized. In other words, in another pathway, de novo synthesis pathway, which is activated ribose, which is being added with the amino acids, and the amino acids and the ATP and carbon dioxide and the other molecules, they contribute to synthesis the base, and then they are adding to it. So this is another way to synthesize a nucleotide. This is salvage pathway. Okay. Let us see on the importance of these amino acids. The importance of nucleic acids now. Why we have to study the nucleotides here and then here. What is the importance? So the importance of this nucleic acid, it can also act as a cancer therapeutic. Cancer therapy or therapeutics. Because the anti-cancer drugs which stop or inhibit the DNA synthesis. So that's why we are interested in studying the nucleic acid biosynthesis in the biomedical importance. And also the next week we are going to discuss about the drug development, another chapter, I think the last chapter in 36, I think, and then followed by you have final exam the following week. So I'd like to concentrate a little bit on the drug development, how the drug design which is going on. That will be useful for you if you want to go for any pharmaceutical or biotech industries. So this chapter will be useful. Okay. So the DNA synthesis, where it will inhibit the DNA synthesis, it can inhibit the DNA synthesis as a synthesis of DNA precursors. Where the precursors is coming from? The precursors are coming from nucleotides. So as I mentioned earlier, the nucleotides involve different components, different amino acids, or carbon dioxide, or ATP, and then ribose. So all of them combine together. They have to join in a particular way to synthesize the nucleotides. So if you inhibit in any one of those pathways of amino acid or carbon dioxide or ATP and all, then you can stop synthesizing the nucleic acid. So in thereby we can design a new therapy for the cancer therapeutics. So we will see some of the excellent therapy, cancer therapy for the last couple of years. We can study using some of the mechanism for the next week. I'm not going into detail today, but we will see next week about that. So the next part is the pathways, which I mentioned earlier. There are pathways, there are two pathways which is involved. Two pathways. These two pathways, one is, as I mentioned earlier, which is focused on the PowerPoint, salvage pathway. Number two, de novo synthesis or de novo pathway. Okay. So what you see in the salvage pathway is the direct addition, direct addition of base. De novo pathway, the base is synthesized. So you should remember these two. What is the difference between the salvage pathway and de novo pathway? Base is synthesized from the basic element like a carbon dioxide plus ATP plus ribose, an activated ribose molecule and other. So, and also the amino acids, So all of them to synthesize this base. So this is de novo. But here the base is available. Is already some of the cells have been recycling, some of the dead cell, or say in some cases, you are eating the food and then vegetables and enough material from meat, from animal protein, so animal tissue. So from the cells of the animals or cells from the plant and other resources from diet, that provides some base for this one, the adenine, guanine and those bases, and that will be used directly synthesis for the salvage pathway, whereas the de novo synthesis is the synthesis of the base itself is synthesized from elements like amino acid, carbon dioxide, ATP and ribose. So you should remember the nucleic acid metabolism or synthesis of nucleic acid or nucleotide. Part of nucleic acid is the nucleotide. The nucleotide is synthesized using two pathways, salvage pathway and de novo synthesis pathway. Okay. The framework of how these pathways being activated, or the framework. Okay. The framework for pyrimidine. Framework of the pyrimidine base is assembled first. First, it is being assembled. Framework. Okay. Then what you can attach ribose. Attach the ribose sugar. Okay. In purine base is synthesis piece by piece. In other words, in the de novo synthesis, purine base is synthesized piece by piece. Piece by piece. That's what individual component. Okay. Directly onto sugar or ribose. So here the pyrimidine is first attached directly to the ribose, and then on a de novo synthesis another one where purine base, that base is being piece by piece individually is being add or attached or directly or to the ribose, and thereby its synthesis. Okay. The de novo pathway again. Synthesis ribonucleotides. Okay. First ribonucleotide is being formed. Ribo. Otherwise RNA is formed. Okay. And then what? From RNA you are producing DNA. Because in the ribonucleotide, when you get the deoxy group, deoxy, if you remove an oxygen molecule into one of the sugar or reduction process, the deoxy means removal of oxygen, addition of hydrogen, that is called here, reduction. So if you see the removal of oxygen of ribonucleotide molecule, so you can change from the RNA to DNA, and this is a major breakthrough on the evolution. In the evolutionary process, in evolution, the theory goes like this. First RNA is being produced, and then from the RNA you get DNA or the other nucleic acids. So you should be very careful when you see the synthesis of from RNA to DNA or DNA to RNA. But in the evolutionary process, they said about the theory is going the first creation of this RNA in the world, RNA world they call it, and then from the RNA the DNA is being formed, and then species and other animals and everything is coming from them. So the all deoxyribonucleoside or nucleotide DNA or deoxyribonucleotides are synthesized from ribonucleotides, and this is being facilitated by that's the removal of oxygen. Okay, and the another one, the difference between the RNA and to DNA Okay, one is the deoxyribonucleic acid, and the other one, which is the thymine. Thymine, which is present only in DNA, not in the RNA. I mean, that is also being performed by the addition of methyl group. Okay, yeah, methyl group is being added, or the methyl group is that distinguishes the thymine of DNA from the uracil. Methyl group is extra. Okay, uracil of RNA. So the RNA contains uracil, and that uracil, if you add a methyl group, and that will become a thymine, and that become a DNA. So how you synthesize the RNA to DNA? You have to deoxy, removal of oxygen, and then deoxyribose group, and then the uracil, from the RNA of uracil, you have to add a methyl group, and then it will be transformed into the DNA molecule. So this is the way that how the RNA and the DNA is being synthesized in a way. And this is one of the picture or the figure. We'll see that one here. There you can see that. This is some of the pathway, the salvage pathway and de novo pathway. Bicarbonate, and you have ammonia with the ATP carbamyl phosphate. You can see the carbamyl phosphate, the pink color, and then the aspartate is a constitute for the CCCN, the 1 nitrogen and 6th position carbon and 5th position carbon, 4th position carbon, all of them form it here. And then it constitutes a pyrimidine ring formation, and then the PRP here, and then what, UTP to CTP, and that is to RNA, and then the UTP to TTP, that is on the addition of methyl group, and also deoxyribose to DNA. So this is a pathway which you can remember, and it's also in your textbook. This is the mechanism: bicarbonate and the carboxyphosphate and carbamic acids, and the carbamic acid to carbamyl phosphate, the carbamic acid to carbamyl phosphate, and carbamyl phosphate, how that carbamyl phosphate, which is being is been activated in this enzyme. And just I'd like to explain just a little bit here how the pyrimidine ring formation, how the pyrimidine ring is formed. Okay, they are assembled from bicarbonate, aspartic acid, aspartate, and ammonia. Okay, that the glutamine hydrolysis, it will give this one here. So what you really get in this glutamine in the primary source for the amino group by the glutamine hydrolysis, that will produce more of ammonia. Okay, the carbamyl phosphate synthetase, okay, that is the active site for this enzyme that contains cysteine and histidine residues. Okay, what is the enzyme? Carbamyl phosphate synthetase. This is a key enzyme. Why I am concentrating on this enzyme? Because this enzyme, on active group, active site of this enzyme, you get cysteine molecule and histidine molecule, histidine amino acid, So these groups are conserved from generation to generation and after generation to generation. So this enzymes is a common, or this particular active site is a common for bacteria and the human or different organism. So that's why the evolutionary process, the pyrimidine synthesis or pyrimidine ring synthesis, carbamyl phosphate synthetase plays a major role in the evolutionary process. Okay, conserved in the evolution. Now, the figure 25(4), I want to explain. The intermediates can move between the active sites by channeling. This is very interesting phenomena in the nucleic acid biosynthesis. The carbamyl phosphate that synthetase, they contain 3 different active sites. This enzyme which I explained to you earlier, 3 active sites. Okay, so these 3 active sites will exchange, will transfer the product. Suppose this is 1 and this is a 2 and this is a 3. Okay, one enzyme, but you have a 3 active site. But here the product which is formed for this active site, then it is not going out, but it is going to the second active site, and then this is the second active site. From this one, it will go on the third active site. So one enzyme, but you have 3 different active sites, but the substrate and product which is formed from here will not go outside, but it will move inside the enzyme molecule and then next part the enzyme. So 1, 2, 3. So all this position, it will transfer, the product will transfer. And this process which is called as a channeling. In other words, the substrate channeling, substrate. So the first substrate for this, and then for the second active site, the product of the first active site become the substrate for the second active site, and the product of the second active site become the product of the third active site. So in total, we call it as the substrate channeling. So you should remember the carbamyl phosphate synthetase is a very sensitive enzyme, and they have channeling of substrate, and without the product going out of the enzyme, out of the cell. So if it goes outside, the product or the intermediate will be inactive. It is not no more active or degraded product, or the reaction which is so fast, within seconds these changes should occur. So this is the unique process when compared to the other enzyme. The other enzymes, if you have the enzyme substrate, the substrate will bind and then it will give the product will go. So the product will come out of this enzyme from the active site. But in this case it is not. It is getting inside the enzyme and inside the another active site, and then it will come out. Okay. So what is the reason for that one, or the benefit for the pathway? Why we have to study? What is the benefit of the channel? Benefit. That one should remember. What is the benefit for this pathway? Okay. Prevention of the intermediate loss. So prevention of this intermediate is so sensitive, and the intermediate will be losing intermediates, the substrate intermediates, because of diffusion process. Suppose that particular and suppose this is the substrate, it will comes out, it will, because of the higher concentration to lower concentration, more inside the cell. Suppose inside the cell, this is the enzyme, this product which is coming out, so it will diffuse outside the cell. So it will not get the end product, it may go out. So instead of going out, it's being channeled inside. So that's the prevention of intermediate diffusion, number 1. Number 2, the benefit is the labile intermediate. The intermediate are so labile, so they are, suppose the carbamic acid, which is so labile and so unstable, it will decompose within 1 second. 1 second. As soon as synthesis, it will be out. So it should be used for the next active site. Otherwise, it will be gone. Okay, and the physiological pH. And the transfer from one active site to the another, in your textbook they have given 18 angstrom or 35 angstrom or 47 angstrom. These are the distance from one active site to the other. So within that active site, this within a second, this compound should be moved to the next one, so that it will be, next step will be added to it. So this is the channeling of substrates. Okay. So now the next part is the orotate acquires ribose. Okay. Another intermediate on the synthesis. Orotate acquires. Okay. ACQUIRES, acquires a ribose molecule, a ribose ring from PRP, from phosphorybyl pyrophosphate, PRP, to form pyrimidine ring. Pyrimidine ring. Okay. So the one which I mentioned earlier is the carbamic acid or carbamyl phosphate. Coming to the PowerPoint again. So these are this one here: glutamate hydrolysis site, bicarbonate phosphorylation, carbamic acid phosphorylation site, and all of them here. This enzyme. Now you can see the glutamine, ammonia to carbamic acid, carbamic phosphate to carbamyl phosphate. Could you see the channeling process? Here the ammonia is formed. Immediately it should go to the carbamic acid, and immediately it should go into the carbamyl phosphate. If this ammonia is going outside, it will be diffused out. So it is not going outside the enzyme, but inside the enzyme there is a channel. That is called the substrate channel. Could you see the carbamyl phosphate synthesis, a red color here, and then here the carbamic acid is the blue in color, and then here glutamine is, you have a dark brown color over here. So this is the active site 1, active site 2, and active site 3. Okay, this is the mechanism of the carbamyl phosphate, and then finally carbamyl phosphate and dihydroorotate. So carbamyl phosphate with aspartate, the orotate is being synthesized. So the ring is being completed here. So what you get onto carbamyl phosphate, the ring basic structure, and then aspartate combined together to form the green color aspartate that contributes the individual atoms to synthesis, carbamyl aspartate and carbamyl aspartate to orotate. And then the ribose phosphate, PRPP, phosphoribose pyrophosphate synthase, okay, that enzyme. And now the orotidylate, and then is combined together to form uridylate, and the uridylate form orotate, and then you can write uridylatate synthesis. You can see the structure, synthesis of the intermediate metabolism, and then UTP, and to form CTP. How the CTP is being synthesized from UTP. Okay, there's a glutamine and ammonia. Ammonia is necessary for CTP reaction. And this is the overall mechanism where you can see this one: carbon dioxide, red, and then N-formyl tetrahydrofolate contribute this carbon, aspartate contribute this nitrogen, glutamine contribute this nitrogen, glycine contribute this, all the yellow CC and N7, N10 formyl tetrahydrofolate contribute this carbon 8, and then glutamine is contribute for the 9, and then ribose here. This is the purine ring structure, and then you have IMPs been synthesized, that inosyl monophosphate from this ATP synthesis, the ATP to DATP, and then GTP, DGTP, and then if this constitute ATP, GTP towards RNA synthesis, and DATP, DGTP synthesis for the DNA synthesis. So these are the nucleotide synthesis in a different way that contribute our de novo synthesis. That individual atoms or individual molecule contribute the synthesis of the ring, that is de novo purine ring structure. Okay. And this is again the same mechanism of the phosphoribosylamine and phosphorylation and the mechanism. The enzymes of the de novo synthesis of purine synthesis, you have glycinamide, ribonucleotide, GAR. Here you can have a transformylase. These are different steps, okay, that constitute the de novo purine biosynthesis. Okay. And this is the overall reaction, how the IMP is being synthesized. And you can go for the colored atom, the molecule, so that individual involved, and also involve the channeling process. This is the split up, that the reaction which I showed you earlier. This is the individual steps for the IMP reaction. Cyanate, and then AMP and GMP is being formed finally. Okay, that pyrimidine synthesis. So the nucleotide monodi triphosphates are the interconvertible. That's you should remember from here. Okay, the nucleotide like a mono or a di or tri phosphates group. Okay, nucleotide, they're interconvertible. Interconversion. What is interconversion? Here, like a UMP, okay, uracil monophosphate plus ATP molecule, which will give you a UDP molecule, because ATP will provide a phosphate group to form the monophosphate, become a diphosphate group, UDP, and it become an ATP group plus ADP group, a diphosphate. So monophosphate 1 and 3 phosphate, 3 plus 1, 4, but here 2 plus 2, 2 phosphate here, 2 phosphate here. So this is purely of the interconversion of these nucleotides. So this plays a major role to synthesis the one nucleotide into the other nucleotide. Also, I mentioned earlier the salvage pathway. Salvage pathway is very direct. Use of already made base, right? There is no individual assembly, but the base is already available. The base is adenine, guanine, thymine, uracil, and cytosine. They are available from diet, okay? And thereby, or any other resource from plant. The diet is from the plant or animal or bacteria or fungi or any biological organisms where you have a life, which have a DNA, which use those meat or any other fruits or anything. From that, you can get the ATGU and everything, and that being directly used onto the ribose, and thereby salvage pathway is very simple, and you make use of your own dietary resources. The part is very important is the thymidylate, okay? The thymine plus you have a deoxyribose D-phosphate, deoxyribose 1-phosphate, and it forms thymidine plus inorganic phosphate. And this thymidine with the ATP molecule, it will form with use of thymidine kinase. Thymidine kinase, it will produce TMP plus what? ADP, which I mentioned earlier. A monophosphate is being added, and you have a diphosphate because the ATP. You have ADP is the product, and TMP is being synthesized. And this TMP is very, very important in the synthesis of DNA. Okay, so thymidylate kinase or thymidine kinase, it plays a major role, and synthesis of DNA. When you get the DNA synthesis is being increased whenever there is a cell replication. Cell replication, you get more of DNA synthesis. That means more of thymidine or TMP synthesis. That's what you need. If you want more, you have more enzyme. More enzyme of what? More enzyme of thymidine kinase. So this enzyme level which fluctuate. In fact, the S phase of the cell cycle, the synthesis phase, right? In the cell cycle, you have a S phase, and then you have intermediate resting phase cycle, and then you have a G1 phase and G2 phase. Like here, the S phase of the replication, cell replication in a cell cycle, you get more of this enzyme, or the more of the thymidine kinase enzyme will be available in the mammalian enzyme. One more thing. This particular enzyme, thymidine kinase, the thymidine kinase is also present in virus as well as in the humans or animals or the mammalian system. Here it is a different kind of kinase, the thymidine T kinase, I put it, thymidine kinase enzyme. Here it is different. Thymidine kinase is different. So if you design an inhibitor which will block only the inhibitor will block here, block the synthesis of or the activity of the virus thymidine kinase, that will not touch here. In thereby you can synthesize or design a drug. Design a drug or antiviral drug, which is being available, and we can also study on the different target, for therapeutic target, which is also we will also get in the next 2 week lecture on the drug design. So one of the drug design strategy is suppose if you want to kill a virus or kill a bacteria or kill unwanted cancer cell. So you have to identify the unique enzyme of the virus or bacteria or the cancer cell, and then thereby you have to design a drug to inhibit this individual pathway, and thereby you are not inducing a toxic effect to the host, toxic effect. So reduce the toxicity of the patient or the host where you are treating those patients. So this is one of the strategy for designing the drug. So based upon this, there is a drug for the herpes simplex virus. Herpes. Have you ever heard of the herpes simplex virus infection? This is being treated by ACYCLOVIR. Okay. Acyclovir. So this is a thymidine kinase inhibitor of virus, not for the human. So thereby this drug is good for the antiviral therapy. Okay, to stop this DNA synthesis of this viral. So that is a unique, different kind of enzyme, thymidine kinase. So what I'll do, I'll stop here, and then I will give you a break for 10 minutes, and then we will go ahead with the other type of synthesis into the product. Okay, nucleic acid synthesis. So in the figure in your textbook 25.5, we have studied the origin of atom, right, of the individual ring. Origin of atoms of that molecule, nucleotide molecules. Okay. And the salvage pathways, they have economized the intracellular energy expenditure. Importance of salvage pathway. Why the salvage pathway is economically feasible or economically advantage when compared to de novo pathway. De novo require lot of energy, require ATP energy to synthesis the molecule, and the salvage pathway is a recycle, right? It can it is because it is available from the food and other pathways, so it is more of advantage of that. Okay. Now let's go into the degradations. Metabolism means, we have to synthesis as well as the degradation of these nucleotides, and the degradation of this the nucleotide or these pathways, which involve different type of diseases that we will see in a short while. But the adenine, the salvage pathway, the two enzymes which we have seen in the salvage pathway, there are two enzymes is important. Okay, so number one, adenine phosphoribosyl transferase, adenine phosphoribosyl transferase, and this catalyzes the formation of AMP. This forms for AMP. This is very important in salvage pathway. And the another one is the hypoxanthine. Hypoxanthine. Okay. Same, the enzyme name, I'm writing it here. Guanine phosphoribosyl transferase enzyme. We got HGPRT. Okay, in short, we call it as the HGPRT. Hypoxanthine guanine phosphoribosyl transferase enzyme. And then here's the adenine phosphoribosyl transferase enzyme. They are very important in the salvage pathway for what? AMPs, call them purine biosynthesis. Okay. So in any case, there is a mutation which is occurring here, HGPRT enzyme, or here. What will happen? You will not be able to get more of purine synthesis. Not meaning a DNA synthesis. Not means you cannot get the DNA synthesis for the the cell growth or cell division or the replication process. So people, they are aiming to inhibit this particular enzyme. Then they found out difficulties and other pathways which is aiding to synthesis this, and this drug designing is not that much successful. But in other words, they have a side effect too, and there are some inborn errors of metabolism of this that we will see in a while now. So what will happen? The HGPRT, this enzyme, it will HGPRT enzyme, and it involved in the formation of what? GMP, okay, and IMP, GMP and inosine monophosphate, guanosine monophosphate, okay, and they are the precursor for GTP and ATP. Okay, precursor for GTP are guanylate and adenylate. Okay, so these enzymes again in the salvage pathway of purine is very important. Okay, that's what I want to stress it now. Okay, the next one, the deoxyribonucleotide. Deoxyribonucleotide, and this is being derived from where? It is synthesized from, from where? Reduction of ribonucleotide, reduction reaction. Reduction means removal of oxygen, meaning reduction or addition of hydrogen. That is also called reduction. Reduction of ribonucleic acid, ribonucleotide. So deoxyribonucleotide is synthesized from ribonucleotide by reduction, okay, or the removal of oxygen. So that's the use on that. And this process, there's an enzyme called reductase. What reductase? Ribonucleotide reductase enzyme. This enzyme will convert RNA into DNA. Okay, ribonucleic acid to ribonucleotide from this one to the, it will help. And it also consists of, this particular enzyme is being clearly, or the mechanism is clearly done by E. coli. E. coli enzyme, they have the same conservative and same type of reaction which is also happening in the mammalian cell. So E. coli is a model, and they have identified, they have genetically engineered this particular enzyme. They found out two subunits involved, two subunits, like a R1 subunit and R2 subunits, and which you can see in your textbook figure. I'll go through that. So this is how the inosinate, adenylosuccinate and fumarate and adenylate AMP and GMP formed from INP. So that's the pathway. And here you can see the how this is an electron micrograph, the photomicrograph of the cells, which is being shown. This is the nucleic acids, and then how they are, the accumulation of the labeling of this nucleic acid synthesis pathway. Okay. On the cell, hypoxanthine, inosinate, and then here how the ADP, GDP, CDP, ribonucleotide reductase. That's what I was just talking about here. Ribonucleotide reductase in the RNA or DNA and the nucleotide. Then how they are being processed into DATP, DGTP, DCTP, TTP. Okay, that's the reaction. And this is involved in the two subunits, R1 dimer, R2 dimer, and then how these active sites are being involved in this reaction. Tyrosyl radical site, tyrosine radical which is involved to synthesis. Again, the same thing, how the tyrosine radical is being formed on this conversion of reductase reaction. And this is again the enzyme. The yellow is the enzyme polypeptide, and then the red and then here how this reaction is being facilitated. They have done clearly with a different enzyme structure. And the same second, third step, fourth reaction, fifth reaction, sixth reaction. How they've been synthesized. Now, the final one, ribose unit to deoxyribo unit, okay, NADPH and NAD. So these are the electron transfer, different reaction where thioredoxin reductase, and then here the ribonucleotide reductase RR here. So you can see this reaction, how this one unit is being converted into this. So this is the final reaction, but to activate this reaction, this should be converted to this. And then this is recycle to get the thymidine, and this is the NADPH to NADPH. So this is the thioredoxin reductase and thioredoxin T, and then ribonucleotide reductase. So these are the total mechanism of reaction, how the ribose unit is converted to deoxyribose unit. So that's very interesting. Okay, this is thymidylate enzyme, thymidylate, dihydrofolate, and different steps which is involved in the, how the thymidylate is being synthesized. Okay, this is very important step, thymidylate. As I mentioned earlier, anti-cancer drug which is being aiming to stop the synthesis. Dihydrofolate to tetrahydrofolate. So the enzyme dihydrofolate reductase enzyme, which I showed you in the earlier class on the dihydrofolate reductase enzyme, by methotrexate is an inhibitor for this, and thereby you can stop synthesizing the DNA intermediate, the thymidylate. Fluorouracil, this is again an anti-cancer drug which will just like a uracil, but the fluorouracil which we inhibit, the suicide inhibitor, fluorodeoxyuracilate, that inhibit the thymidylate synthase, DUMP, DTMP, and thereby dihydrofolate is being inhibited. But in the aminopterin and methotrexate, they inhibit this dihydrofolate reductase enzyme. So either you can inhibit thymidylate synthase, or you can inhibit this. In either way, you are going to stop the tetrahydrofolate formation, where the glycine serine, where the methylene donor and everything, all of them which is being stopped. So these are some of the drug designing strategy which you can apply here for the cancer, anti-cancer drugs. And this is the fluorodeoxyuracilate. This is another mechanism how this enzyme is being stopped on the stable adduct. aminopterin, or is the H, or methotrexate here. This is another compound. Trimethoprim. Okay. And how this particular reaction is being regulated. This is very important by the feedback mechanism, right? Feedback mechanism. How this enzyme, the synthesis is being stopped. When you see the aspartate carbamyloid phosphate, here the ATCase, aspartate transcarbamylase transferase reaction. Okay, aspartate. Okay, transcarbamylase, aspartate transcarbamylase enzyme. That is called the ATCase enzyme. This one, AT and case enzyme. This enzyme is sensitive for the product of the CTP. The CTP, if more CTP is synthesis, the CTP will go on inhibit this. At the same time, if you have more energy, you can activate this process to CTP reaction, and thereby you can synthesis. So in an energetic point of view, if you reduce the ATP reaction or the ATP synthesis or the ATP energy cycle, or if you consume less carbohydrate, less fat, where you reduce the synthesis of overall energy production in your system, you can reduce the chance of getting cancer. In other words, the cancer prevention strategy, they said reduced calorie. This is the way the reduced calorie will work out, by reducing the synthesis of this, and thereby what? It will remove, or it will reduce synthesis of the CTP, and thereby, even though if there is a mutation or even though there is a carcinogen, but if you reduce the calorie, people they think that okay, we can reduce the cancer formation or the cancer carcinogenesis. So this is the feedback inhibition, and that's again a strategy. And the purine biosynthesis is controlled by feedback in several sites. This is the next one. See here the pyrimidine synthesis. Here the inhibited by IMP, AMP, GMP, phosphoribosylamine. Several sites, not one but several sites. This is again the purine biosynthesis is being inhibited. Okay. Now the next enzyme, the biomedical importance of that is ribonucleotide reductase enzyme. The ribonucleotide reductase enzyme, which I mentioned earlier, this is a control point or checkpoint, control, regulation. Okay, if you disrupt this, the nucleotide metabolism, if any disruption of this metabolism, that will lead to pathological conditions, and the adenosine deaminase reaction, adenosine deaminase reaction, it will, the loss of this enzyme, loss or inhibition of adenosine deaminase reaction, that will lead to several severe combined immunodeficiency. Severe combined, COMBIND, combined immunodeficiency. See, that means the immune reactions is being so badly regulated. That means you are suffering from SCID, S-C-I-D, meaning the person is always suffering from the immunological disorder, like a SCID disease. In other words, have you ever seen the bubble boy disease? Bubble boy disease? So you cannot live for a long time because of the loss of thymus-derived cell, T cell, loss of T cell. So that means the immune system is totally switched off. So the boy won't survive because of the external, there's no immune system. So you have to survive these existing systems, which is the innate immune system, not for antibody systems about it. In other words, in the another type of reactions which we can study about the gout, G-O-U-T, gout, is another disease that induced by the high level of urate, urate crystal or urate, in the high level of urate in blood as well as in urine. How the urate is formed? That is the metabolic product of the inosine generated adenosine deaminase, then to what? Hypoxanthine. Hypoxanthine become xanthine. So hypoxanthine converted to xanthine by xanthine oxidase. Xanthine oxidase. So this enzyme, xanthine oxidase, produces more of xanthine, and then the xanthine with the other oxidation, that will lead to uric acid. And this uric acid, the loss of proton as phosphorylator, the physiological pH, the uric acid, which will form with the other atom like a urate crystal or urate. In normal urine, if the excretory product of purine, the purine of the nucleotide, right? The purine, adenine, guanine, right? So they will eliminate as urate in the normal process. Suppose if you get excess the metabolism which is going on in the system, the urate will be more, and this urate will again precipitate in kidney, forming the stone formation, or on joints in a base of the fingers like this, or here, or the base of this finger, joints, it accumulates in the membranes, and thereby it crystallizes, crystal formation. The crystals like a needle. So if anyone is suffering from gout, like a needle prick, onto that joint, it's a heavy, painful onto that. So anyone is suffering from this, that's exactly of the gout. In some occasions, the uric acid is crystallized. Excess of some people, they cannot tolerate spinach or some green vegetables which they contain urate crystals, that also contribute the urate crystal. So you should avoid. And urate is again the deficiency of these enzymes, aminodeaminase enzyme. And the treatment for this particular disease, the gout, allopurinol, allopurinol, allopurinol. This is again an interesting molecule. It is an analog of hypoxanthine. Hypoxanthine, like a competitive inhibitor, as well as it will inhibit that enzyme xanthine oxidase. Let us see that one here. And the xanthine, the hypoxanthine, to xanthine. So allopurinol is a substrate. Allopurinol, I'll write it here. Allopurinol is a substrate, and as well as an inhibitor. Substrate as well as the inhibitor for what? Which enzyme? Xanthine oxidase. This is an interesting one. Xanthine oxidase enzyme. So if you inhibit this enzyme, you will not get more of uric acid, so urate is not being produced. So this is very highly active, and the urate or uric acid, which is something like ascorbic acid, like a vitamin C, it's a highly scavenging molecule, or reactive oxygen species scavenging, or antioxidant property, this uric acid. So what people think, if you have more of these uric acids in the blood, that can also protect, or for the longevity in for the long live. If you want to live for long, if you have more uric acids, and that can promote removal of oxygen, reactive oxygen species, and thereby it will promote the health. Also, they can also reduce the incidence of cancer. So that is another way they are also thinking, because highly accumulating of the metabolic products towards the uric acids, crystals on the urate which is coming out of your body from the normal is the excess amount. That is a very quite a reasonable amount of metabolism or the catabolism of purine in terms of the uric acid, and thereby the cancer incident will be reduced. Okay, that's another point. And the another one which we have to remember is the HGPRT enzyme, which I mentioned earlier, and hypoxanthine and guanine, and then phosphoribosyltransferase transferase enzyme. And this pathway, there's a mutation, which is a recessive disorder, and this mutation of this in a salvage pathway, this will lead to self-destructive behavior. Self-destructive behavior, or this particular person or the child always biting the nail, or sometimes biting their fingers. So this, if you leave it for a while without restraining, they will eat all these fingers or chew the lips. So it's a very dangerous situation, self-destructive. Okay, and they also suffered from psychiatric disorder, like a mental disorder. So you should be very careful for this particular Lish-Nihan syndrome. So the people who suffered from this call it as a LYSCH, Lish-Nihan syndrome. Have you ever heard of this one, Lish-Nihan syndrome? Inborn errors of metabolism. Okay. And in other words, they also called as a compulsive self-destructive behavior. Compulsive self-destructive behavior. Okay. They also behave aggressively towards others. So aggressive person. And mental deficiency, they were elevated urate in kidney for this, and kidney stones is more common. And also this is a sex-linked recessive disorder. Sex-linked recessive disorder. This particular disease. And the another one, normally we call it as a folic acid deficiency, in vitamin C deficiency, like a folic acid. It's also vitamin, right? By folic acid deficiency. This also leads to a disease called spina bifida. Spina bifida. Have you ever heard of this one? They have the neural tubule formation, the early stages of the neural tubule formation in the pregnancy. Okay, during pregnant women, and there is one case in 1,000 childbirth, they suffered from this disorder, a spina bifida. There's the incomplete formation of neural tubules. Incomplete formation of neural tubule. Okay, that's an early development. So the people, they used to, if they want to have a child, so they have to take before and after, or during the first trimester. They have to take lot of folic acid to avoid the disorder of incomplete neural tubule formation, and thereby they can promote of DNA synthesis. Why? Because during the pregnancy or the first trimester, the DNA synthesis or the nucleic acid synthesis is highly elevated. At the time, you need to supplement this folic acid, which is very necessary for the tetrahydrofolate or dihydrofolate, or from the urate to thymidine synthesis, which is necessary for the DNA synthesis. So for this, you need a large amount of folic acid or folate. So that's why the folic acid that will help. If there is a deficiency, then the neural tubule formation is being defective, and thereby the people, they suffered from what is called spina bifida here. So these are some of the clinical importance of the biomedical importance of this particular topic of nucleic acid synthesis. Do you have any questions so far? Do you have any question? Sugarland? No. Here, Victoria? No?
No, sir.
Cinco Ranch? No.